Yes, so today we presented the results from our phase 1 clinical trial using CD5-directed autologous CAR T-cells to treat patients with relapsed refractory T-cell malignancies. This was a first-in-man study using a CD28 zeta CD5-directed CAR T-cell for patients who had either relapsed following chemotherapy or transplant or had primary refractory disease to chemotherapy or relapse following prior chemotherapy. A group of very heavily pretreated patients. We had a total of 11 patients that were actually infused with the product, 6 with T-cell lymphoma and 5 with T-ALL, and those were the results that we presented today. So to date, we've treated 11 patients on study, 5 patients with T-ALL and 6 patients with T-cell lymphoma, and we saw striking results in the lymphoma cohort with a 50% overall response rate. Of those 4 patients, 1 patient was able to proceed to allogeneic stem cell transplant and remains in remission 9 months out. 1 other patient has been in remission for 7 months without need of additional therapy. And another patient remained in remission for 7 months before unfortunately relapsing and requiring additional therapy, but remains alive and in remission at this time. We had 1 response in T-ALL patients and a pediatric patient, which unfortunately was a short-lived response, and she ultimately progressed. And so the next steps that we plan to take are dose escalation. We've completed dose levels 1 and 2 to date, and we'll be enrolling on dose level 3 soon. And we hope that by boosting the dose, we will be able to also boost potency of the CAR T-cells. We also are looking into some additional manufacturing changes so that we can try to boost the efficacy of the CAR T-cells as well. In addition, we are working on developing a donor-derived product for patients that have relapsed post-allogeneic stem cell transplant so that we can start with the healthier T-cell population. And eventually our ultimate goal is to develop an off-the-shelf platform where we can treat patients immediately without need of waiting for production of the CAR T-cells. Based on the 11 patients that we've treated to date, that appears to be the case for us as well. We did have 1 response that occurred on dose level 1, with the remaining 3 responses occurring on dose level 3. So the numbers are still small, but again, as we escalate doses, we do hope to see more responses, but it may not necessarily be correlative. I'd just like to say that it's an exciting time to be in the field of hematology, particularly cell and gene therapy. There's so many amazing presentations that are being given just at this meeting, particularly around CAR T-cell therapy with new constructs that are being tested, as well as a lot of presentations on mechanisms of resistance and how we might potentially overcome these mechanisms so that we can extend this therapy to a broader range of patients in the future.